Welcome. Through this presentation, I would like to give you an overview of our new public-private partnership initiative, G-Force. This project will focus on identifying natural enhancers of GLP-1 and other appetite-regulating hormones, as well as exploring how pharmacological GLP-1 treatment may alter food preferences. Please note that what you will see today is an early outline of our idea. If the topic is of interest, we warmly invite you to get in touch. Your suggestions and input are very welcome as we further shape this project. In recent years, we've seen a major shift in the treatment of obesity, largely driven by the emergence of the pharmacological GLP-1 receptor agonist such as Ozempic, originally developed for type 2 diabetes. These drugs have proven remarkably effective in promoting weight loss, often with significant improvement in metabolic health. This is transforming the way we think about obesity management. At the same time, it raises important concerns and new questions. We also see new opportunities scientifically. What about natural enhancers, mechanisms, and changes in food preferences? So, as mentioned, the rise of pharmacological GLP-1 agonists comes with questions, concerns, but also opportunities. First of all, we know that many consumers have aversions to a pharmacological intervention. They are also not available for everybody because the treatment is expensive. And what about the side effects, the effects on the long term? For instance, it's a basic concept that administering high doses of a compound might reduce the body's own production, and this might partially explain the gastrointestinal side effects observed. This provides sufficient arguments for exploring natural enhancers of GLP-1 and other satiety markers. Another important question that arises for us relates to food preferences. As more and more people are being treated with pharmacological GLP-1 agonists, we are particularly interested whether, beyond the regular reduction in appetite, there are specific types of food that becomes less appealing. For example, imagine if such a treatment leads to a reduction in protein intake. That is likely not the intended outcome, and it could have an important implication for the nutritional status on the long term. These are precisely the aspects we're aimed to investigate in this project. So in this project, we aim to explore natural dietary strategies to stimulate the release of GLP-1 and other satiety hormones. We seek to better understand their mechanisms of action, and importantly, we also want to investigate whether pharmacological treatment affects food preferences. So within G-Force, we will identify natural ingredients that can enhance GLP-1 release and better understand their mechanisms. Partners can supply ingredients that we will test. We will also find dietary strategies that may prevent overconsumption without pharmacological treatment by identifying the satiating capacity of food items. Lastly, by gaining insights into the pharmacological GLP-1 treatment influence on food preferences, we aim to better understand and respond to the evolving needs of this rapidly growing consumer group. These three aims will be addressed in three separate work packages, but these work packages will be connected with each other. Within Work Package 1, we aim to identify natural ingredients that can enhance GLP-1 release and gain a deeper understanding of the underlying mechanisms. Our focus will be on endogenous GLP-1 production, but we will also examine the release of related gut hormones such as GLP-2, which is derived from the same precursor protein and secreted in parallel, yet has a distinct biological function. In addition, we will assess the role of enzymes such as DPP-4, which degrades GLP-1, and explore whether any ingredient can inhibit this activity to prolong GLP-1 action and other satiety hormones. To achieve these goals, we will use a combination of in vitro and in vivo approaches. We will use cell-based assays to assess the potential of an ingredient and clarify their mechanisms of action. We will build receptor binding assays to screen whether compounds can act as a ligand for the GLP-1 receptor. And we will perform clinical studies to evaluate postprandial GLP-1 responses in humans. And as mentioned earlier, partners can supply their ingredients, which we will include in our testing pipeline. We can test fibers, proteins, rare sugars, or other bioactive compounds. In our second work package, we will quantify these key components found in work package 1. So the fibers, the proteins, rare sugars, and other bioactive compounds. And we will quantify these in selected foods to estimate their satiating potential. We will focus on foods that are particularly relevant for individuals at higher risk of obesity. Are they affordable? What is the context of experiences? How to label them? And are they easy to prepare, for instance? We will also conduct a human study using the most promising food candidates and assess the impact on satiety after repeated exposure. This to better reflect real-life dietary patterns and potential long-term prevention effects. Finally, in the third work package, we aim to gain insights into how pharmacological GLP-1 treatment influences food preferences. Understanding these shifts is essential to better respond to the evolving needs of this rapidly growing consumer group. We are particularly interested in whether the appetite-reducing effects of GLP-1 agonists apply uniformly across the diet or whether they selectively impact scientific food groups or macronutrients. For example, protein, fat, or sugar intake. This knowledge could have important implications for long-term nutritional adequacy and the development of supportive dietary strategies. In Work Package 3, we will make use and leverage on an existing cohort that we're building in Wageningen. In this cohort, we follow individuals undergoing obesity treatment. A subset of these people will receive pharmacological intervention with GLP-1 agonists, such as Ozempic. To assess the impact of treatment on dietary choices, we will measure food preferences before and after the intervention using comprehensive food frequency questionnaire and appetite-related assessments. To carry out this project, we have assembled a multidisciplinary team with expertise in cell and receptor assays, human clinical trials, and specialists in consumer behavior and satiety. To truly make an impact, we believe collaboration with industry is essential. That's why we're inviting partners who want to stay ahead in the rapidly evolving field of GLP-1 and satiety. Whether you're supplying ingredients, developing food products, or manufacturing end products, this project offers you the opportunity to explore the potential of your innovations in a scientific, robust setting. Importantly, we also aim to understand the needs of a new and fast-growing consumer group, individuals using GLP-1 agonists. Their food preferences may shift significantly, and being able to anticipate and respond to that could be a real competitive advantage. To conclude the main part of the presentation, a few additional slides will follow with some general background information. We're very open to suggestions on how to strengthen this project further, and we welcome ideas on how to align with the interest and the expertise of potential partners. Please don't hesitate to reach out if you see opportunities to collaborate. Thank you for your attention.